And I'll just use my same kind of generic virus that we drew over there. So here's my nucleic acid. Here's my, what's this, you guys? My capsid, right? To make this an envelope virus, I'm going to coat it in stolen host cell membrane. So here's the envelope. Here's the viral envelope. And if I ask you, folks, what is the source of the viral envelope, what will you tell me? What is its source? Where did the virus get the envelope? Yeah, it's stolen host cell membrane. It could be cytoplasmic membrane. It could be nuclear membrane. Some viruses steal membranes from the endoplasmic reticulum. So the viral envelope then is stolen host cell membrane. Now the question is, folks, would it make sense for the virus to have the adhesins attached to the capsid? Would this make sense? Why doesn't that make sense? Yeah, the envelope is covering the adhesin, so the adhesin can attach the host cell surface receptor. So if you were designing this virus, well, I better be careful. If you were designing this virus, where would you want the adhesins to go? In the envelope, right? So this is a cool thing, you guys. The viruses modify the cell membranes are going to steal as envelope. The viruses modify those stolen host cell membranes by inserting their viral proteins into the membrane before they steal it. And this makes sense because it's in the envelope that the viral adhesins must be located. So the viruses have to stick those protein adhesins in the cell membrane that they're going to steal as their envelope. Okay. So these then would be the envelope viral adhesins. And there can be other viral proteins in the envelope, but we're trying to kind of make the most basic virus that we can. Now, when you do your reading, often these viral adhesins are described as glycoproteins because very often there'll be little sugar residues attached to the proteins. Okay, on a short answer section, if I ask you what are viral adhesins made of, if you just say protein, I'm very happy with that. Good. Okie dokie. So now that we kind of walked through that, now we'll look at some slides and we'll talk about why is it important to know if a virus is naked or enveloped? Who cares, right? It turns out it's really important. Okay, so this is just a cartoon of a generic naked virus. So what's this purple spaghetti inside, you guys? Nucleic acid, and depending on the virus, it will be what? DNA or RNA. Good. These little balls are the protein subunits called capsomeres, and you can see they self-assemble into the beautiful protein code called a capsid, right? So, and we'll see the capsids, there can be different shapes to them. The most important job, well, there's two most important jobs of the capsid in naked viruses. The first one is to protect the genetic information from destruction, and the second one is, and we can't really see them here, you guys. We drew little spikes here. What would those little spikes be? The viral adhesins, right? Now, in some viruses, they do have little spike-like structures as adhesins. But other viruses, the adhesins might be very subtle. They might not be beautiful spikes. The viral adhesins could be just really subtle little proteins on the surface of the capsid like that. But making the spikes is always more fun. This is so important, you guys. Big stars, big stars, big stars. The bad news for us humans, if we're dealing with an outbreak of a viral pathogen that's naked, is that naked viruses can remain infectious in the environment for long, long, long periods of time. And the reason is this outermost coat is this tough, resistant protein capsid, and it's on this tough, strong capsid that the viral adhesins are located, right? So naked viruses, when they're shed into the environment, they can remain infectious for many, many, many days, maybe weeks. And that's bad news for us humans, right? Because that means that potentially we could become infected from a naked virus that's just in the environment. A great example, you guys, polio virus. The virus that causes polio is a naked virus. It's shed in fecal material. Bad news for us humans. Since it's a naked virus, once it gets shed in feces, it can remain infectious for days and days and days. So wherever the feces goes, if it contaminates food or drinking water, and then we swallow that contaminated food or water, even if the poliovirus has been sitting out for days, it's still infectious, right? We can still get infected. Bad news for us, these doggone naked viruses. Here's some examples of some naked viruses and the damage they can cause. This picture maybe was from the 1940s or 1950s. These are mechanical ventilators. They were called iron lungs. So it turns out, luckily, a small percentage of folks that get infected with poliovirus, the virus can invade the central nervous system and cause paralysis of voluntary skeletal muscles, including the muscles involved in respiration. So these poor folks that have suffered from polio, to survive, to breathe, they were put in these iron lungs. And the iron lungs go through cycles of positive pressure, negative pressure, so it passively causes the chest to move in and out and passively causes the ability to breathe. But can you imagine, you guys, you're stuck there. You're stuck there. And they'd have wards and wards and wards of people in iron lungs. Luckily, in the 1960s, yeah, no, I'm getting 1960s, late 1950s, 1960s, we had 2 polio vaccines developed. So that's why nowadays, at least in parts of the world where we are able to vaccinate our children against polio, we don't see hospitals filled with people in iron lungs. Okay, and in the RNA section, you guys will come back and talk about polio. It's a naked RNA virus, fecal oral transmission. Another virus we're going to discuss is a DNA virus. This is the human papillomavirus that causes so-called warts. And I remember as a kid, like we go to the public swimming pool, and my mom would say, make sure you wear your tongs or your calypsos or your sandals, otherwise you'll get warts. And as so often happens, your mom was right. Okay, so HPV is a naked DNA virus. It can cause these growths that we refer to as warts. So this person, for example, if they went to the pool, they would be shedding naked wart viruses onto the cement, maybe in the changing room. So if you then came along and you had maybe a little bit of damage to the bottom of your feet, that's a great place for the HPV virus to invade your skin, and then you become infected, and then you would have these wonderful plantar warts, right? So again, the disadvantage for us humans, since the HPV, they're naked viruses, they can't survive for long periods of time. Survive is probably not a good word. They can remain infectious in the environment for long periods of time. Just another one really quickly is a virus that can cause relatively mild upper respiratory tract infections that we refer to as colds. One of the cold viruses is called rhinovirus, and it too is a naked virus. And this explains why in wintertime, when a lot of us have colds, respiratory tract infections, let's say I have one and I'm going, achoo, achoo, achoo. So there goes those naked viruses into my hand. So when I'm going like this, and I'm going like this, and I'm touching the door handles, I'm contaminating those surfaces with that naked rhinovirus, which then can remain infectious for many, many days, even after I'm gone. Yeah, Michelle.
How well does disinfectant like Lysols kill those? Because like you see on the Lysol can, it says kill 99.9% of the germ virus.
Yeah.
Do they kill viruses?
They probably do. If you think about it, with the naked viruses, anything that causes protein denaturation, all you need to do is denature those little protein adhesins, and that virus is no longer infectious. So the old formulations of Lysol had phenol in it, and phenol is a denaturing agent. The newer formulations don't. So it's always good to look at the label and see if it says specifically, will this antimicrobial, will it inactivate lipid, they sometimes call them lipid envelope viruses, will it inactivate naked viruses? But again, just anything that denatures proteins, is going to denature the adhesins and make the virus non-infectious. Yeah, so we're going to be doing, well, you guys know that. We've already done this in a lab with bacteria, but we can maybe explore a little bit more about those same antimicrobials we've tested in lab against viruses. Maybe I can get a little deeper together for you guys. Because this controlled microbial growth, you guys, I know a lot of times it's really boring, but in the real world, that is so important as healthcare providers. knowing what will inhibit or kill the microbes when you're dealing with patients that have infectious diseases, it becomes so important. I remember in school, I was like, this is so boring, all these chemicals. But then when you get out in the real world and you're dealing with an infectious disease outbreak, that information is so important to you. Okay, onward. The next group of viruses, as we said, are the envelope viruses. So again, here's a generic one. Just so you know, folks, in this particular envelope virus, the capsid is in what's called a helical shape. We have a helical capsid. And I know it's hard to see here, but kind of this pinkish ribbon, that would be the viral genetic information. And we see the viral protein capsimers. They bind to the nucleic acid. And it creates kind of a spiral staircase helical capsid here. But what's new is this outermost layer. So what's this outermost layer called? This is the viral envelope. Good. And what's the source? Stolen host cell membrane. Good, you guys. And what are these little lollipops? Yeah, these are viral glycoproteins, and we would presume that in this picture, they must be what? What function? The viral adhesins, dynamite. Okay. If we have an outbreak of a viral infectious disease, you guys, we are praying that the virus will be enveloped. And the reason is, where are the adhesins located? In the envelope, right? So anything that damages the envelope will make the adhesins non-functional. Now, if you can remember back to chapter 4, we were talking about cytoplasmic cell membranes. We were talking about the fluid mosaic model. And you remember how we said the consistency of cell membranes is almost like olive oil? How delicate that is, right? It's a very delicate structure. So the good news for us humans is if we're dealing with an outbreak of viral pathogens and the viruses have an envelope, once they're shed from their host into the environment, the envelope can quickly be damaged simply by drying, by sunlight, by lipid solvent chemicals like soaps. Anything that damages that delicate envelope will destroy the ability of the adhesins to function and make that virus non-infectious. So a specific example, the only good news, you guys, about HIV, what do you think? What's the only good news about HIV? It's an envelope virus, right? And this is why that when people freak out, when HIV was first discovered, it was horrific because people were like, I don't want you to be in the same room with somebody that has HIV because I could get it from touching something they touched. That's absolutely baloney, right? So as soon as the HIV is shed from the host, and this is important, folks, once it dries up, it's no longer infectious. Now, where you have to be careful is envelope viruses. If they're protected in organic material, like tissue, like if it was a huge blood spill, and the protective tissue, the protective organic material remains moist, that will protect the envelope. Okay, so I mean, we have masses of organic matter. That provides a protective fort for envelope viruses. But if it's like a tiny drop of blood and it dries up, that HIV is probably going to be inactivated, within maybe a few hours. Just another example of how you need to watch out for organic material protecting envelope viruses. When we talk about the influenza virus, it's an RNA virus, it's enveloped. We're going to talk about one of the groups called avian influenza viruses, bird influenza viruses. And what's really intriguing is that in birds, avian influenza viruses replicate in the intestinal tract. So what would we find large numbers of avian influenza virus? What excretion of the bird? Feces, right? So the bad news is, you guys, they've done studies on duration of infectivity in avian influenza viruses in moist bird feces, and they found that in moist bird feces, avian influenza can remain infectious for up to 3 months. So that just tells us if the envelope virus is in this protected, moist, organic material, they can remain infectious, right? So just want to let you know, and we'll talk about this more, because if avian influenza ever hits California, we're on this Pacific Flyway where lots of wild birds pass through. We want to remember that to protect our children, Because I know when our kids were little, we loved to go to the park and feed the ducks and the geese, and there's bird feces everywhere, right? And you're tracking it home on your shoes, and your kids are rolling in it. Yeah, so we just want to remember that if we ever get avian influenza here in California, we'll have to be really careful about duck and pigeon and goose poop. Okay. So a couple of the envelope viruses we'll be discussing. A DNA envelope virus, herpes viruses, we'll be discussing next week. And as we mentioned, an RNA virus, HIV, we'll be discussing next week as well. And again, you just want to remember, the good news about these envelope viruses, they do not remain infectious in the environment for long as they're not protected by moist organic material. We'll talk about structure of viruses. We just want to, again, mention the size of viruses. This size range, you guys, it depends which textbook you're using. I mean, I was taught the virus size range is about, for most, 30 to 300 nanometers. Just so you have an idea, we're in this ballpark, 30 nanometers to 300 nanometers. This resource said 10 nanometers to 400 nanometers. The important thing is, as we mentioned, is for light microscopes, about the smallest thing we can visualize using a light microscope is around 200 nanometers. Okay. So most viruses are smaller than 200 nanometers. And again, this is why we need our light microscope. Just to give some comparisons, if this is a cartoon of a human red blood cell and erythrocyte at about 7.5 micrometers, maybe to 8 micrometers, here's a typical bacterium like E. coli, about 3 micrometers by 1 micrometer. And what do you think these little dots are? These are viruses, okay, just to give us an idea. So we're going to zoom in here just to look at the sizes of some viruses. And again, I don't expect you to memorize the specific sizes, but know the unit of measurement when we're describing size of viruses is what? What's the unit of measurement? Nanometers, right? In bacteria, what's the unit of measurement we use to describe micrometers, right? So we're talking about like 1,000-fold difference, 100 to 1,000-fold difference. So just to get you oriented, you guys, here's the wall of the E. coli. And this, whatever reason, this nerdy bit of information I cling to. So the bacterial ribosomes are about 25 nanometers. And what I remember is, okay, the size of a ribosome is about the same size as the smallest viruses, which are the polioviruses. So see how here they're saying bacterial ribosomes 25 nanometers, polioviruses around 30 nanometers. I can remember that the smallest viruses are about the size of ribosomes. And so you can imagine, you guys, when you're looking at your bacteria in your light microscopes, can you see the ribosomes? No, way too small, right? So that helps me remember why in most viruses we can't visualize. Here are two different bacterial virus families or examples. The bacteriophage T4, which we'll be exploring in more detail in a little bit. This is another family of bacterial viruses, bacteriophage MS2. And again, we just see the differences in size. Here is one of the larger viruses, members of the pox virus family. The pox viruses are some of the largest viruses. And indeed, you guys, with pox virus infections of cells, such as smallpox, we could probably just barely make out a smallpox virus in an infected cell. But again, we're going to say for most of the viruses, we need an electron microscope. Tobacco mosaic virus, we can see here, it's quite long, 300 nanometers, but very, very narrow, 15 nanometers. Now, this part can get a little bit boring, so we'll just get through it. So we do want to remember with regard to describing genetic information of viruses, viruses will use either DNA as genetic information or RNA. So we have DNA viruses and RNA viruses. An important point here, folks, is which enzyme would copy viral DNA? Which enzyme would copy viral DNA? DNA polymerase. Can DNA polymerase proofread? Yes. So consequently, when DNA virus genetic information is being copied, there's relatively low mutation rates, right? Okay, now let's look at RNA viruses like HIV, like influenza virus. Which enzyme copies viral RNA? RNA polymerase. Can RNA polymerases proofread? No. So that means RNA viruses have the potential for really high mutation rates. And that's exactly what we're going to see next week when we talk about influenza viruses and HIV. their mutation rates are so high. And it's for this reason that with influenza viruses, every year we have to get revaccinated because there's so many mutant strains and they're always mutating all the time. We can't develop one influenza vaccine that will protect us against all the mutant strains. It helps us to understand why we still don't have an HIV vaccine. We're going to discover HIV has another step in replication, which is going to introduce even more mutations, right? So because of this high mutation rate, we can't develop an HIV vaccine. It explains why both influenza viruses and HIV, they can rapidly evolve drug resistance. And it explains why our immune system can't ever get caught up, right? Once we get infected with these viruses, let's say with HIV, and we have lifelong infection with HIV. HIV is constantly mutating. Our immune system is trying so hard to keep up, making new antibodies against the new mutant strains. But eventually, the high mutation rate of HIV, it overwhelms our immune system, right? So it's heartbreaking. Okay, viral genetic information can be double-stranded or single-stranded. We can have linear viral genetic information. We can have a single linear strand of viral genetic information or multiple segments, almost like little mini chromosomes. Or some viruses have circular genomes. And in general, the size of the viral genome, it's going to be much smaller than the genomes of cellular organisms. And this is just one example. That little tiny, tiny bacterial virus, the MS2, the genetic information encodes for 3 genes. So we could say 3 proteins, right? In contrast, the smallest cellular organisms, one of the smallest bacteria, the chlamydia, their genetic information encodes for 1,000 genes, or we could say 1,000 different proteins. So we can see, the viruses just don't carry much genetic information, and that's why they rely on their host cells to carry out most of the processes involved in viral replication. And this is a great example of where a picture or photo is worth 1,000 words. I still am just blown away by this. Electron micrograph. This is a single E. coli that's been lysed, releasing, what's all this? Yeah, that's the single chromosomal DNA of this E. coli. Isn't that amazing, you guys? That is craziness. How does it get in there? Supercoiling. Okay. This is the single circular chromosomal DNA of this bacterium. And in contrast, here is the DNA of a bacteriophage. So just physically looking at the difference. And again, this is to help us understand why viruses can only replicate inside a host cell. They don't have enough genetic information to carry out biosynthesis of their viral genetic information, their viral proteins. So they have to parasitize a host cell and use the host cell's biosynthetic machinery to make copies of themselves. Okay, viral shapes, just really quick, you guys. So here's the helical shape. Polygonal shapes are very common, and a really common polygonal shape, I wouldn't ask this on an exam, just so you know, is icosahedral. And icosahedral capsids have 20 triangular faces. There are more complex viruses. Our bacteriophage is an example. In the T phages that we looked at earlier, this is called the head. This portion is called the tail. The head is the capsid protecting DNA. The tail, we're going to discover, the tail is used for attachment to host cells, and it's used to inject host cells with the phage DNA. Another example of complex viruses, meaning that they're made of more than just genetic information, a protein code, and an envelope, are variola viruses. The smallpox viruses, these are envelope viruses, but they have additional proteins inside, these lateral bodies. The function, I don't know what the function is. And another complex virus is the bullet-shaped rabies virus. It's complex shape with additional proteins. And again, I haven't done my homework on the function of the additional proteins found in rabies and the smallpox virus. Now, I don't want you to panic, you guys. I'm going to show you a table of DNA viral families, and then the next table is RNA viral families. And we won't be looking at all these families, but just so you know, if you want to use this as a resource, I inserted an N if the virus family is naked. If there's no N, it means they're enveloped. So these are all the DNA virus families. We'll be talking about herpes viruses and papilloma viruses and hepatoviruses next week. But what I do want us to take note of is the number of DNA virus families. So we've got 1, 2, 3, 4, 5, 6, 7. So 7 DNA virus families. And now let's take a look at RNA virus families. Wow, a lot. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. More than twice as many RNA virus families as DNA virus families. Any suggestion, you guys, why? Why do you think we see greater diversity amongst the RNA viruses? Yeah, you nailed it, right? Remember, they're mutating. This really high potential for mutation. So that the consequence of that is that we have much greater diversity in the RNA viruses. So again, twice as many RNA virus families. Next, what we're going to do, you guys, and I hope we can get through this, is we're going to use your viral replication cartoon from last time, and we're going to talk about, it is a model for next week, is a model for us to understand how is it that viruses invade our cells and replicate. We're going to use the historical route. We're going to take a look at the first viruses where the replication cycle was understood. And that's the bacteriophages, the bacterial viruses. And you might say, well, why did people study human viruses first? Remember, viruses are often in intracellular parasites. So in the old days, to study viral replication, you'd have to infect humans, right? And we know, although in the past, well, nowadays, hopefully we know that's not ethical, right? So as humans, we think it is ethical to infect bacteria, right? There's no lobbies to protect bacteria. So most of the early work was done in bacteria. And it also is an advantage because bacteria replicate so quickly and the phage replicates so quickly. So you can study many, many generations of bacterial viruses in the lab within a few weeks, right? So many advantages. And again, there are no ethical issues killing bacteria while studying these viruses. So we're going to take a look at viral replication. And we'll do a little concept map here. For in viral, bacterial virus replication, there's 2 basic cycles. And the 2 cycles are called the lytic cycle and the lysogenic cycle. I know, it is just, oh, it's going to get exciting next week, guys. I tell ya. You will be sitting on the edge of your seats when we start talking about human virus replication. Okay, so we're going to look at bacteriophage, replication cycles. And what we're going to do is we're going to look at 2 different families of bacteriophage. Okay, to look at these 2 different types of replication, we're going to look at 2 different bacteriophage. And the first group are called the so-called lytic, or some folks call them virulent phage. And the reason is they can only carry out the lytic replication cycle. And we'll see in the lytic replication cycle, they always kill, they always kill their host by lysis, by lysing them so virulently, causing severe disease.